We will take a further step to our next presenter, who is Professor Szekely from Sweden, from Karolinska University. Professor Szekely has been living in Sweden for decades, and he's a real innovator. And I'm really grateful for his initiatives. He has wide experience, almost globally, I could say, with China, with other countries, in Indonesia, how to implement therapeutics and diagnostics. He's a real developer, and he is also fond of developing this region by applying techniques and giving instruments to our region to test tumors. With this, I would like to ask Professor Szekely to give his keynote lecture, which we are looking very much forward to as well.
Thank you very much. I hope that you hear me. I'm just testing the technique through this setup I haven't tested before. So I like to point out the sentence what Martin said, that testing is more and chancing is not favorable in cancer. And my first slide to show that I'm very much favor of testing live tumors and their reactions for the different treatment options. So I'm coming from Karolinska Institute. My original professorship is in hematological malignancies, and we are the ones who are giving the Nobel Prize. And I'm also working as a practicing pathologist responsible for autopsies, rapid autopsies on cancer patients, and also high-risk autopsies. And I'm also responsible for molecular testing at Karolinska. So just a very brief overview that the reason why we trust that cancer has to be treated in a personalized way is coming from the notion that cancer is a genetic disease. And the underpinning for this is the famous experiment of Peter, who showed that you can transmit cancer using cell-free extract from chicken fibrosarcoma. And this transmission is done through recombinant retroviruses, which are picking up cellular genes which are incorporated in the virus genome. And using this technique, on the second part of the 20th century, up to 70 different oncogenes were identified, and they showed to be members of the signal transduction pathway, but where the cells is sensing the environment for growth promoting signals and transmitting these signals to the nucleus to change gene expression patterns. The other group of genes which we are regularly working in, both in the clinics and in cancer research, are tumor suppressor genes identified with the help of small DNA tumor viruses, which code for sequences which are needed to bring a resting cell to a dividing cell state. And these viral proteins are interacting with cellular partners, the two famous group of p53 group and the pocket protein group of retinoblastoma. And these genes are responsible for controlling the integrity of the genome, like p53 is sensing all the DNA damages and induce either cell growth arrest if the damage level is low, or induce cell death if the damage level is high. And this gene has to be eliminated frequently during tumor progression. And the changes in the p53 system can be also beautifully monitored in different animal systems where mutations in the tumor suppressor gene actually brings high tumor frequencies, mirroring the human familiar cancer syndrome, Lee syndrome. The other heritable tumor is retinoblastoma, where the identification of breakpoints at chromosome 13 and cloning the genes from the breakpoint revealed the mechanism of the disease initiation. So now we have over 70 oncogenes, close to 100 tumor suppressor genes, and a number of tumor modifying genes. All of them are forming more and more complex regulatory networks. And all these sort of mathematical precision description of the tumor generation from the very first step until the metastatic tumor gave us a great hope that now cancer is something that we can simply, rationally fight. We just identify the mutations and find the targeted drugs which are aiming for these changed proteins, and always matching the therapy to the identified mutation. And we are still kind of living in this nice dream, but time to time we start to wake up. So our daily diagnostic procedure now involves gathering of a huge amount of multi-omics data. So we sequence many of the genes, either a small set of critical genes for a particular disease, or 500 gene sets for most of the driver mutations, all whole genomes or whole exomes, combined with transcriptomics, combined with metabolomics and proteomics. And we hope that gathering more and more data, we will be more efficient in dealing with the disease. And this is our normal working procedure. We obtain the sample, get the multi-omics, and then sit down in the tumor board. And as long as we find mutations which are fitting nicely to the repertoire of the targeted drugs, we are happy. But more and more often we get frustrated because you have the number of variants of unknown significance. You have the combination of low frequency alleles with each other, where we actually do not have any real idea. So basically, if we have a targeted drug matching the mutation, we use that for the treatment. And this treatment can be very impressive, proper treatment. Let's say if you have BEMORA family, identify a BRAF 600 mutation, it gives you spectacular first response. But many of our patients actually do not have a sustained response, and often relapse, and they don't live actually longer than the traditionally treated, but they have a nice period of intensive drug response. Actually, I think more than 60% of the drugs which were approved since 2012 have no increased overall survival effects when people tested them in the post-marketing surveys. And our daily life is that we have certain tumor types where genetic testing is now routine. For example, EGFR testing in lung carcinomas, where roughly 1 out of 6 Caucasian actually have targetable EGFR mutation. And if you, at the same time you identify bypassing mutations like in the ROS or RAF, you can have elimination of the effect of the EGFR pathway. And you can also identify a number of less frequent mutations which has their own targeting medication. And continuous testing also can reveal variations in the mutation pattern. Some of them are actually making the tumor resistant for the therapy. So we test a lot. We find occasionally targeting drugs, but the tumor is sort of evolving constantly under the selective pressure. And for many cases, we do not cannot provide new therapeutic options. And the reason why it's so difficult to treat this, that if we now, when we can sequence all the genome, and we have now the history of many 10,000s of total genome sequence, we realize that most human cancers have a staggeringly high number of mutations, 3,000 to 50,000. Of course, it's relatively few of them are what we consider either targetable or driver mutations. And when we look at the frequency of mutations, which can be actionable, we have this trouble with the long tail curves. So we have relatively infrequent mutations in very large number. So we have p53, ROS, PI3 kinase, which are regularly coming up as a mutant, but many of the drive mutations are in the 1 to 5% range. And it is further complicated by the fact that if we start to do molecular excavations at different areas of the tumor, or compare the mutation pattern from different metastases at different places, but also over the time, we will start to get a great heterogeneity. So tumor is extremely plastic, extremely rapidly evolving, generating new variants if we put a selective pressure on it. The other Scrooge, what we really realize is that our diagnostic categories are not necessarily describing the biological feature of the tumor. So traditionally, we are associated with organ anatomic location-based and histological subclassification, which we consider as one entity in the treatment. Detailed transcriptomics analysis, when people just calculate the principal component of the individual transcriptomic pattern, which basically describes the biological entity of the tumor, shows that different tumors have a very much overlapping pattern. So you can have a colon carcinoma, which is more similar to a breast carcinoma than to an endocolon cancer. So the question is that when we identify our target groups and we try to treat them, and because this is the basic of all drug approval procedure, do we get the proper target groups? And we can also illustrate it in our everyday work. If we just, for example, take lung cancer and we stain them with 30 different markers, we see the patterns emerging which are corresponding to the histological subgroups, but also we see that there are basically no two tumors which are identical for many of those diagnostic antibodies what we use in the predicting of how shall we treat them. So when we get to the personalized therapy, this is one of the greatest paradoxes what I used to point out, that in modern medicine, everything has to be evidence-based. But when we are talking about individualized therapy, we don't really talk about individualized. We talk about a person who belongs to a certain cohort. We select out one or few diagnostic markers, and we say that those individuals who have these diagnostic markers, they belong to a group, and historically evidence show that a certain treatment is working on them. Is it good enough? So today we have around 140 approved drugs. If I just looking at the small molecules in the EU, ever increasing number of immunological agents, biological agents, and so on. And many of them are surprisingly effective in a minority of the cases. So some of the most effective drugs what we have, they are extremely promiscuous. So a minority of common tumors are nicely reacting to it. And this one is complicating very much the picture. And also at the same time, we have hundreds of new drugs just getting out from different drug companies, from different lab sources. So the therapeutic arsenal what we can use are now approaching close to 1,000. So now we get back to the classical question: how can we find the best drug for an individual patient? And the old dream was that we can imitate the principles of the antibiotic testing on bacteria. So if we manage to get the tumors in a ex vivo live culture, expose them to drug, and we just see the effect and use this as a guiding principle. And this is over 40 years old dream. Generations of postdocs spent many of the years on it, and then some of them were successful, so the technique is slowly improving. And what we realized that the challenge is not so to run this assay effectively, the challenge is not conceptual, it is purely practical. And the real challenge: can we keep the primary tumor cells outside of the body, not just alive but happy, so that for sufficiently long time to test the drug effect. And I would like to point out that in our view, the most important is that when you remove the tumor cell from the body, the tumor cell should not even experience that actually it left its original source, and it should up as soon as possible in an environment which is similar to the environment the cell is experiencing inside the body, meaning that actually it should be a quite hypoxic environment and quite reducing environment. So we generated culture systems like this where we can nicely keep the tumor cells alive up to 9 days. And our focus is to isolate tumor cells which can be growing in suspensions or as in small 3D structures, what we often experience in different effusion tumors. So late-stage tumors where tumor cells are shedding into the pleural effusion or ascites. We also work with hematological malignancies which can be isolated from blood or lymph nodes. And to isolate them, often we just use simple physicochemical methods like gradient centrifugation and then removal of non-tumor cells using adhesive methods or different dynamics. To make these assays highly reproducible and affordable, we adopted a format where we use 384 web plates with standard 30 drugs, one single plate, and 5 different dilutions. So first concentration is the one which is corresponding to the dose what the patient receives, and then 5 times 25 times 125 times, and we repeat it 3 times. And now our assay includes 150 drugs. Basically, we cover the entire spectrum of pharmacological solution which is available today, either as a prescription drug or drugs which are in the phase 3 tests anywhere in the world. And our aim is to generate this is a highly reproducible, industry standard, affordable system. So we have automated microscope which is reading these plates, photographing every well, and counting the cells. And we also use high precision pipetting robots. And we generate the plates using acoustic nano technology, where we can produce 700 plates from one single master plate. And the result is something like this, that after the incubation with the different drugs, we have a solution where we fix, kill, permeabilize, and quench the fluorescent signal in one single step. And this renders the cells for a countable system. And if the drug is working, we see that higher concentration is eliminating the cells. If drugs have no effect, cell number doesn't change in the proportion of drug concentration. And we do it in triplicates. And these are the list of the 150 drugs what we right now using. Beginning of next year, we plan to extend it in 240 drugs, where we will also include combinations of the most frequently used therapeutic protocols. And when you look at the effects, so we count the cells, we count them with 5 different counting algorithms, so that independently the cell is a single cell push transron or growing in 3D aggregates, we get a reliable counting, and we generate the drug response charts. And to make it sure that you can combine them, we use a simple statistical method, a weighted sum, where we actually count the decrease of the cell number in proportion to the logarithm of the dilution, because drug response curves are logarithmic. And we normalize it to 1 to 100. And because we use those concentration ranges which are approved for the particular drug, it makes it possible to compare drug effects with each other. Even if some of the cases, it's a gram quantity drug or microgram quantity. So we have this as an automated system, and now we try to set up a number of hospitals, and we are running clinical trials both for hematological patients and solid tumors. So when it comes to solid tumors, we use those tumors where you can easily generate cell suspensions. So, for example, hepatocellular carcinomas, kidney cancers, where a simple scraping of the tumor surface yields suspendable cell aggregates, which are possible to pipette, then we can effectively measure drug effects. In those cases where there is an extracellular matrix, we use a different technique, what we call solid sense, but I will not talk about a lot about it. But what I just like to point out that this technique is suitable for cases where basically you exhausted all the therapeutic options. So if we take a patient with lung cancer who shows treatable mutation in EGFR, which is later disappearing after the therapy, and the patient is going through traditional chemotherapy, then new mutations appears which are possible to treat, and now tumor is spreading to different places, including the brain. It's generating a large tumor mass in the mediastinum, which is radiated. And then finally, a number of mutations shows up, which makes now the targeted treatment ineffective. And this is the time when we can come in and now isolate cells from effusion and identify the best effective drugs from the full spectrum of available chemicals, and still give a chance for the patients for treatment. So this was one of the first part, and I would like to look a little bit in the crystal ball, how ordinary pathology actually will function soon, because the techniques what we use today are still just at the late 1900s or 1800s level. So we take pieces from the tissue and we take a thin slide and stain them with a textile dye and the bark extract, and we just get stainings of the different shades of blue and red. And nowadays we digitize them, but it's still just a telepathology. It's not really digital pathology yet. And then we generate number of pictures and have an army of pathologists who spend a great deal of their life to recognize these patterns and recognize the normal patterns, and then eventually identify the pathological patterns. And now with the rise of AI, we hope that we could decrease this challenge for the human eye and human brain and replace it with the computer. So we try to train the computer up, for example, to identify tumor cells in lymph nodes. And if you now spend a lot of time, a lot of specialist doctor training options to make all the annotations where you identify different cell types in the lymph node and also identify tumors. That's in this study we had 70,000 annotations, and then you can train up the system that it will be effectively identified just by looking at it, the presence of colorectal tumor. But this is something what the pathologist could do just immediately in a few seconds looking at the slide. But we are still insisting on it that we should sort of use the computer technology to identify cases which are rather difficult and often rather questionable for human interpreter. And we kind of refuse to get extra information. We're still sticking to the textile dye and the bark extract staining to actually solve some rather mundane job, identify where the matrix or the stroma is compared to the tumors. And this is an interesting challenge, because if something humans are very well versed and selected for during the revolution is to render a concept to very sketchy visual information. So we are trying to challenge our computation system to do the things what humans actually doing absolutely best. So are there any alternatives? So we run a lot of high-scale immunostainings, and we realize that when you meet a new pathological situation, for example, when you look at COVID lungs and you say that you want to get all the information out of it, you can make a series of immune staining or series of sections and expose the slides to a number of immune staining, describing actually basically all the interesting biological processes. So in this case, I just showed 30 different antibodies in combinations with each other. And then we realized that if you use enough antibodies, you could cover most of the human pathologies. So we estimate that around 95% of pathological challenges could be described using this set of antibodies. Now the question is, can you do a massive multiplexing on this. And one of the lessons what we learn is that introduction of the multicolor flow cytometry in hematology made a huge revolution. And it went very rapidly. The technology was not even fully matured when it already reached the routine testing diagnostic laboratories. And just getting more information allowed the classification of hematological changes very rapidly. So can we do this on pathological slides? And for that, if you want to do fluorescence staining, because that's what you can effectively combine with each other, the classical way is that you combine dyes with different emission and excitation peaks, and then you can have dyes which are shifted in position to each other, and you can get a long list of dyes which are now covering a full spectrum, and in theory gives you a possibility to multiplex them. And then you can get systems where you now identify the different cell types. Now, how do you do it when you want to discriminate many different colors? You can use different field test setups. You can use spectral imaging with spectral detectors. And you can also use mathematical tricks to separate the different emissions using emission fingerprinting. And then you have to set up the staining itself. And the one which is now most frequently used in different laboratories use a number of antibodies, label them with barcoded DNA pieces, use the barcoded DNA piece to attach the proper fluorochrome, and then you take batches of 4 or 5 antibodies, do a sequential capturing, and then you stitch together all those images, which gives you beautiful publications. But on the other hand, it takes you 2 or 3 days for one single slide. So anyone is working in routine laboratory, you know that this is not something what you could adopt the clinical use. If you cannot make a full imaging within 10 minutes, the method is dead. But on the other hand, the method provides us already with the analytic possibilities. So how to cluster these cells, how to identify the different phenotypes, and how to look for relations between the individual cells, relations between individual histological borders. Now there is a great development in the technology, because now we have fluorescent markers which can be excited with the very same light, so a single excitation light. They are not bleaching. And now if you have a scanner which can actually do a parallel scanning, so simultaneously scan all the colors at the same time, now actually we are within the realm to get let's say 10 color labeled pathological slide, which can be imaged in the same way as you get the signals from the multicolor flow in hematology. And now we get to the point how to actually feed proper pathological data to AI. So normally this is the way how you use capture, so identify that you are human or a robot. So you give sort of impossible objects to identify for a robot. It takes a huge amount of learning time until you can find out all the different boats. But if you just simply can give a separate color to the boat, a separate color to the bridge, separate color to the cars, then actually AI can do the job what it's supposed to do, namely identify disease conditions based on those definitions, what we use today also to describe a tumor. So instead of using black box clusterings and teaching, and where the results very much dependent on the quality of the ingoing material, now you can use a logical AI where you know exactly what criteria are used to define the pathological condition. So I just make 2 predictions what I'm pretty sure that it will happen, so that if we want to improve individualized cancer therapy and make it affordable, so we can introduce ex vivo assays with high throughput, covering basically all the treatment modalities. And this can be more, not just small molecules, it can be also antibodies, it can be also effector cells, sensitized T cell clones, and also it can be extended to physicochemical methods like oxygen tension, like radiation sensitivity. And this can also be a very effective way to bring down treatment costs, because when we look at the 150 different drugs, what we have, some of them cost $20 per treatment, some of them cost $200,000. And out of the 150 drugs, we have 84 generica. So even on the most stringent economic condition, you can, with a good chance, find a few drugs for basically every economic levels. And my prediction for AI pathology, so to do the diagnostic, is that we will do high throughput scanning with automatically with multiple immunological marker up to 30, 40. And with that, AI will be automatically identifying the disease condition. And this will eventually kill pathology as we know it. So in our future dream, the pathologist who is looking at hematoxylin and staining will be in the same category as those who are repairing Xerox machines or lighting up candles on the street for lighting. So thank you very much, and I hope that I provoke you a little bit. So I'm happy to take questions.
Thank you so much, Professor Szekely. I like your optimistic ending of concluding about the future of pathology. As you yourself, you are also a pathologist, I know. So the question is not what will happen, but how do you predict when it will happen?
I think this, because all the conditions are already ready. So testing ex vivo tumor cells, this is now a large-scale endeavor in several Chinese universities with this technology what we developed. For multiplexing the pathological slides, basically all the components are in place. What you need is a high efficiency scanner, and that one is already past the level of feasibility. So the major reason why these things can happen much faster than we would expect is that many of the components, the electronic components price is sort of dropping like a piece of stone. So CCD cameras, which were basically unaffordable for many years ago, now you can have 10 of them built in a system. So I'm very much optimistic that technology will catch up with this, but not necessarily in sclerotic areas, perhaps in areas which are more industrious and daring in their investment in medical technology development.
Fantastic, thank you so much. Regarding these instruments and testing of such cases, do you have this in the everyday practice? Do you have patient experience by testing ex vivo cases? And I know you work on this for a long time, but how do you see the perspective in the next year for this? You offered an instrument to be installed in
Hungary, yes.
Central Europe, Eastern European region. So how do you see this application in direct patient care?
So the idea is that this can be set up as a franchise model. We can provide the mediums, the ready printed plates, the scanners, the fluid handling pipette system, and also analysis of the scanned images. And so we would like to sort of expand this exponentially and reach many of different places of the world. Our ambition is that it doesn't matter if you are in Shanghai or in a small city hospital of Indonesia. We would like to reach basically every oncology or actually pathology department where you can employ these techniques. And because we show the results of the killing directly on single cell level, so your confidence in the drug effect is not restricted to look at curves, but you look at the real effect of the drug.
Do you see any application in China already?
Yeah, so we have started in Peking Shenzhen Hospital number 2 in Shenzhen with the oncology and hematology group. We run a clinical trial in Guangzhou. which is aiming to get the Chinese IVD application, and we have contract with number of firms who actually want to use this technique for patient pre-selection for clinical trials using their experimental drugs. So we have an constantly increasing group of hospitals who want to adopt this technique. And now, so we produce all the components, and we are setting up the high capacity cloud computation to handle the increasing amount of data, so that we don't just run computers in the lab, but also can adopt potentially thousands or 10,000 cases per day.
Thank you so much. So for your ongoing efforts and decades of implementations and innovation, and also developing this region, we would like to welcome you as an honorary member of the Central and Eastern European Academy of Oncology. I will give you this certification in person or send it to you.
Thank you. It's a great pleasure to be with like-minded people. Thank you very much. It's a great honor.
And thank you for all your input and sharing the knowledge and innovating and taking this our humble world further on. Thank you so much for your nice presentation and attending your precious time. Thank you so much, Professor.
Thank you very much.